Now, properties of genetic material, DNA versus RNA. Till now, what we have studied, search was started for genetic material. Griffith has given the transforming principle. Then every McCarthy MacLeod told that it might be the DNA. And Hershey and Chase has given the proof that yes, genetic material is DNA. So till now, it is clear that the debate between protein versus DNA is now resolved by Hershey-Chase experiment, and it is clear that it is a DNA which acts as a genetic material. It is now clear. But in some virus, you are finding RNA as a genetic material. Example, tobacco mosaic virus, QB bacteriophage. Now, this is important question. Reason is Hershey-Chase has used bacteriophage to prove genetic material is DNA. But QB bacteriophage is having genetic material as RNA. Very, very important question. Please learn it. QB bacteriophage genetic material is RNA. Now, question is asked, why DNA is a predominant genetic material? Why RNA? Next question is, why RNA perform the dynamic function? It is performing the function as working as messenger RNA. It is working as adapter, means transfer RNA. So, if you see, the difference is there because of the difference between the chemical structure of the two nucleic acid molecule. So what are the chemical structure? You know very well. What is the difference between DNA and RNA? First of all, DNA will use deoxyribose sugar. It will use deoxyribose sugar, and RNA will use ribose sugar. Then RNA will use uracil, and DNA will have thymine in place of uracil. Then DNA is double strand and RNA is single strand. So a molecule which can act as a genetic material must fulfill the following criteria. Number one, it should be able to generate its replica. Earlier, again, we are taking the debate between the three persons, that is protein, DNA, and RNA. And what we are finding, DNA can make its own copy by the process of replication. Okay, and because of complementary base pairing, it is able to make its own copy. Even RNA can make its copy through DNA. It can get it done because of the complementary. So if you see, because of the rule of base pairing and complementary, both the nucleic acids have a ability to direct their duplication. We are understanding the first criteria right now. Problem came with the protein. Protein failed to fulfill the first criteria only. So protein was out of the race because produce its own replica is a feature which is shown only by DNA and RNA, and they are able to manage. Why? Because of the rule of base pairing and because of complementary. Now, second criteria was it should be chemically and structurally stable. I'm using the color difference. Second criteria was it should be structurally stable and chemically stable. Stability is one of the important properties which was evident from Griffith experiment, that even after heat which killed the bacteria was killed, but it does not destroy some of the properties of genetic material. So it means stability is really very important property which is clearly seen from the Griffith experiment, because heat which killed bacteria was not able to destroy some of the properties of the genetic material. Clear? So genetic material should be stable enough not to change with the different stage of life cycle. It is not key in the childhood the DNA is different or in the old age the DNA different. No, it should not change with the different stages of life cycle, with the age, or with the change in the physiology of an organism. So it should be stable. Now, how can you explain it? First is, this is explained in the light of that DNA first property is DNA is two-strand. And because they are complementary, if suppose you separate them by heating, they can come together if appropriate conditions are provided. The first important point for explanation is because the DNA is made up of two strands, both the strands are complementary, and if suppose heat is given, temporarily they will separate, they will move apart because they are held by their hydrogen bond. So temporarily they will move apart, but when the conditions are removed, means heat is removed, they will come back. So they come together when appropriate condition come. The first point is easy. Now go to the second point. Second point is second position OH group present in every nucleotide in RNA. RNA has got at second position OH group. So RNA has second position OH group in its sugar is actually a reactive group. Second OH is a reactive group because of which RNA becomes more labile and easily degradable. So please learn the question language if they ask which is a reactive group in RNA, second OH. What is the problem with this group? It is easily degradable. Why? Because RNA is labile. Then main next point, I'm highlighting with a different color, please focus. Then third point is RNA, because it is catalytic, it is reactive. Now question language is what is reactive group and why RNA is reactive? The reactive group is second OH and RNA is catalytic, that is why it is reactive. So conclusion is what? Conclusion is that DNA is chemically less reactive and it is structurally more stable as compared to RNA. So DNA is chemically less reactive and structurally more stable. So out of this, which is the best genetic material? DNA. Clear? Then next, again, this is again going for the second point only. We are right now discussing second point, stability. Now come problem why RNA is less stable. Reason is in RNA, uracil is present. But in DNA, thymine is present, which gives stability to the DNA. So I'll just show you 1 minute how to read this paragraph. This entire paragraph, this two paragraph are for the number 2 point. Number 2 point. We are saying chemically and structurally stable. So we are telling stability is important. It should not change with the age, stages of life cycle of physiology. Griffith experiment also tell heat killed bacteria but does not destroy genetic material. Then DNA, if it is heated, the two strands, because of complementary, hydrogen bond will break because of heat. But if heat is removed, they will come back. Second, RNA is having a second OH reactive group, which is making RNA liable and easily degradable. Third, RNA is working as a catalyst, so it is reactive also. And DNA is stable. Why? Because it contains thymine in place of uracil. So I think so this much paragraph is clear. This paragraph, first paragraph is for the number 1 point. Okay, first paragraph is for number 1 point and second and third paragraph is of number 2 point. So just see this much, are you comfortable or not? And then now I'll move to the next paragraph. That is now third point. It should provide the scope for slow mutation which are required for evolution. And now come to the fourth paragraph. Both are able to mutate. But RNA, because it is unstable, mutated faster rate. That is why the virus which are having RNA as a genetic material, they have short lifespan and new strain of virus come very fast. Now, last point I'm using with this color. It should be able to express itself in the form of Mendel character. So if you see, RNA can directly make protein, hence easily express the character. DNA has to depend on RNA for protein synthesis. DNA has to depend on RNA for protein synthesis. So it means protein synthesizing machinery has evolved around RNA. So from here, what it is clear? It is clear that both RNA and DNA both can function as genetic material, but DNA is more stable. It is preferred for storage, and for transmission, RNA is a better choice. So this is actually a very, very important point for your competitive exam or for your board exam. Each and every word of this paragraph is actually very important. Please go through it. If you have any doubt, then ask. Now, next is RNA world. Now, an immediate question come in the mind, which is the first genetic material? Actually, RNA was the first genetic material. RNA was the first genetic material. And we are having sufficient evidence also to suggest that life process, essential life process like metabolism, translation means making of protein, splicing, we'll be studying in the further topics. All work is done by RNA only. And you know very well, RNA was used as a genetic material by some viruses. And it was working as a catalyst also because it is helping in making the peptide bond formation. Okay. So if you know many living biochemical reactions, though we say that majority are catalyzed by protein, but some still are there which are catalyzed by RNA. So we know that RNA, because it is catalyst, is very reactive and it is unstable. So because of this nature, was reactive. So DNA has evolved from RNA with chemical modification. And you know which chemical modification? From second position, OH group was removed and it became double strand, which make it more stable. DNA is double strand and it is complementary. So it resists changes by evolving the process of repair also. It will do the process of repair if any mistake is there at the time of replication, so that mutation or changes should not occur much. So just read up till here if you have any. Now, replication. Replication, you know very well, DNA will make DNA. So Watson and Crick, when they were proposing double helix model, that time only they proposed a scheme for replication. And according to their wordings, what was this? It has not escaped to our notice that the specific pairing which we have postulated will suggest the possible copying mechanism for genetic material. It was this given by the Watson and Crick. So this is the original statement given by them. According to this scheme, the two strands of DNA will separate and they will act as a template so that new strands can be made on it. After the completion of replication, each DNA molecule will have one parental and one new. So it means DNA replication is semi-conservative. If you see, parents are having red color and a new blue color is made. So as a result, this daughter will have one strand of parent and one new they have made. So they are able to save only half of their parent strand, which is semi-conservative. So who has proposed this model? This was already proposed by Watson and Crick at the time when they were giving the double helix model. So now we have to study the experimental proof. It is now proven that DNA replicates semi-conservatively. It was shown in E. coli. E. coli it was shown, and even in higher plants also, like higher organisms like higher plants and human cell, it is also shown. Who has done this work? It was done by Michelson and Starr. What they have done with the bacteria, they were growing bacteria in E. coli containing N15. What is N15? N15 is a heavy isotope of nitrogen. Please understand it is not a radioactive isotope. Please, it is not a radioactive, it is heavy isotope. So N15 is heavy isotope of nitrogen and this is normal nitrogen, and you can separate both the strands of DNA by which method? Based on the density gradient centrifugation. So this heavy DNA can be distinguished from normal DNA. Normal DNA means N14. Normal DNA means N14 by centrifugation in cesium chloride. Give me 1 minute. By centrifugation in cesium chloride density gradient. So first of all, understand very carefully what they have done. They have grown E. coli in a medium containing N15 as the only nitrogen source for many generation. What is the result? N15 is present in both the strands of DNA. So when both the strands of DNA, they are containing N15, you are calling it as heavy DNA. That is why we are saying this heavy DNA. Now, next, if you are using N14, I'm using black color for N14. So this is your normal DNA. So you can distinguish heavy DNA from normal DNA by centrifugation in cesium chloride by density gradient. First of all, first point, what they did after preparing this E. coli, here, I'm using this red color for N15. First of all, I'll work here. You see this diagram carefully. This is N15, red color. And I'll put them in now N14. So I'm using, this is N14. Okay. This is first generation using N14. What will happen, you know very well, as a result of semi-conservative method, parent will separate and new will be made. And new, I'm using the black color and 14 with black color. So it is made hybrid. Hybrid is made. First generation is over. Now let me go for the next generation. Next generation, again, I'm putting in N14. So what will I do? See the diagram carefully. First, I'll work with this side. So I will separate this black separate. And N14 means clear? Same, I will work this side also. I will separate. So what is the result? If you see my result carefully, this was heavy DNA. These both are hybrid DNA, hybrid, hybrid, and this is hybrid, this is hybrid, and this is light. So simple is, if you put heavy in N14, if you put heavy in N14, what you will get? You will get two hybrid. One heavy will give you two hybrid. Clear? And one hybrid, when you put again in N14, what it will give? It will give one hybrid and one light. Am I right up till here? If
Yes,
looking confused, then please once again see. This was one hybrid, and one hybrid is giving you one hybrid and one light. One hybrid is giving you one hybrid and one light. The two hybrid will give you what? They will give you two hybrid and two light. So after first generation, generation 1, you are getting only hybrid. After second generation, you are getting two hybrid and you are getting two light. These are light and these two are hybrid. And what you are doing it, you are separating them by density gradient centrifugation by using cesium chloride. So if you see the diagram, N15, both strand N15 is at bottom, heavy. When it is hybrid, little up. And when it is light and hybrid, it is light is more up and hybrid is down. So this is the way they have worked and proved that yes, genetic material DNA replication is semi-conservative. Now, one more trick I'll give you. Check here. See here. This is your heavy DNA and I'll use this color. This is your light DNA, means 14 and 14. And this is your hybrid DNA. And I'm drawing 2-time hybrid DNA. This is also hybrid. This is also hybrid. So first of all, understand this is heavy DNA. This is light DNA. This is hybrid and this is hybrid. Now see, when you put heavy DNA in N14, what will happen? They both will become hybrid. They both will become hybrid. Clear? Now, when you put light DNA, in N15, what will happen? N15 means red color. Both will become hybrid. It means if whatever pure they are, if you put in opposite medium, they will become always hybrid. They will always become hybrid when you take any pure and you put them in opposite medium. Now, what will happen if you put hybrid now? If you put hybrid in N14, let us assume put hybrid in N14. So I have to first separate. N14 means I have to use blue color. So what I'm getting one hybrid and one light. If suppose I put hybrid in N15, what will happen? See. I will get one hybrid and I will get one heavy. So what is a learning? Learning tips is if whatever you take hybrid, whatever medium you are putting, one will be of that medium, another will hybrid remain. For example, if you're putting hybrid in N14, one will be light, another will be hybrid. And if you put hybrid in N15, one will be heavy, another will be hybrid. This is my easy learning tips. I hope so this much topic might be easy for you. And numerical question, if I ask you, suppose I ask you, कि Michelson and Stahl experiment is continued for 3 generations. Up to 3 generations, it is continued. 3 generations, this experiment is continued. Means starting from heavy DNA, both N15 and 15, starting from heavy DNA. Then tell me what is the ratio of heavy is to light is to hybrid. So what we have to do, we know one heavy was given to us. After first generation, they became 2 hybrid. In the second generation, 2 hybrid became 2 hybrid and became 2 light. In the third generation, light became 4 light. And this hybrid became again 2 hybrid and 2 light. So total light are 6 light and 2 hybrid. So heavy, what is the answer? Heavy is 0, light will be 2 and hybrid will, light will be 6. Light will be 6 because if you see there are 6 light. So there are 6 light and how many hybrid? Hybrid are only 2. So you will write 2 here or simple you can make it 2. So you can just make it simple 3:1. This is your answer. This question they can ask you. I hope so this much topic might be easy for you to please check it. Now, one more thing, let me explain it here. If they ask for 80 minutes means we have gone for the 4th generation. 4th generation, so they're asking proportion of light and hybrid density. So, if you know till 3 generation, it was 6 light and 2 hybrid. So 6 light will become 12 light and 2 hybrid means again 2 light and 2 hybrid. The total 14 lights will be there. How many? 14 lights will be there. And how many hybrid? 2 hybrid will be there. So answer will come 7:1 ratio. Now, similar experiment was performed by using radioactive thymidine. Radioactive thymidine, okay, to know the newly synthesized DNA, and it was performed on Vishya Faba, who has performed Taylor. This is a scientist and the colleagues in 1958 to prove that yes, genetic material DNA is replicating semi-conservative. So I think so. This much topic might be easy for you. Just read it. If you have any doubt, then ask.